Hello everyone, and welcome to today's SCLF webinar. It's very nice to see you all today. It has been a wee while since we've had one of these in our own rights rather than holding them for someone else, so it's great to be here today. For those who don't know me, my name is Alison Mackay and I'm a member of the SCLF Executive Committee, and I'm one of the organisers today. And I'll be joined by Eve, who's also in the background, and together we'll hopefully make today go nice and smoothly, and if there deal with any sort of technical issues that we might have along the way. If you do have any technical issues, if you just stick them into the questions box, and we'll see if we can try and help you out. Before we get started, just got a couple of housekeeping things to deal with. So you'll have joined today on mute and your camera is off, so you can just sit back and relax and enjoy today's session. And if you do need to step away, then don't worry, you can catch up on our YouTube channel, and I'll put the link for that in the chat later, so you can go ahead there and subscribe to that. If you do have any comments or suggestions as we go through the session today with our two speakers, then just put them into the questions box. And if we don't manage to get to them today live, then we'll try and pick them up offline. And lastly, the housekeeping is just to ask you if you could possibly just fill in the feedback form, which will open up after you close this webinar, and it'll also come into your follow-up email. And you can use that to give us any suggestions of anything that you would like to see going forward in terms of SCLF events and webinars or speakers. Hopefully you'll be able to stick around after this. At 2 o'clock we're going to be having our AGM. I'm hoping it'll just take about 15 minutes. We do need to have a certain number of members there to make it quorate, so it'd be really appreciated if you can stick around for that. I'm not going to say it's going to be riveting, but it's something that we have to do. So we'll get through it as quickly as we can. So hopefully you'll stick around for that. So without further ado, I'm going to introduce our first speaker today. This is Tom's going to have a go again at this. I tried to do this webinar a few months back, and technology failed us, unfortunately, when we tried to do a two-speaker event. So hopefully we'll go better this time. We've got one first this time, so we're hoping for better things. So so Tom Aspray from ERS, and Tom is the technical manager at ERS. And what I'll do is I'll put the full a link to the website, which gives the full bios for both our speakers in the chat as well. And Tom's going to speak to us about the use of qPCR testing in contaminated land projects. So I'll just hand over to you, Tom.
Thanks, Alison, for the invitation, and to the committee as well. Yeah, as mentioned, we hoping it's all going to work technology-wise this time, second time round. So as it's a webinar, there's maybe a few people here that don't know who ERS is, so I'll just very briefly explain who we are. And then I wanted to spend a bit of time talking about what qPCR is. So there may be some people that already know about it, a basic idea of what the technology is. So perhaps for them, it'll be a case of going into a little bit more detail, so that if you're engaging with people that offer that kind of testing, then maybe you'll be able to have a bit more sort of in-depth discussion. And of course, if you're new to qPCR, hopefully I'll give you enough of an insight to feel a little bit more confident about the technique, and then maybe you'll use it in your projects as well. So there'll be a little bit of time spent on that. I'll then sort of say why I think it's a particularly good technology to be using now in contaminated land projects, and then I'll use some case studies to hopefully illustrate that, and those will be case studies from our own projects, as well as providing that as a service to other clients as well, and then we'll just wrap up at that point. So about ERS, I'm not going to go through all the details on this slide. This is a sort of overview of ERS, the clients, and some of the key points about us. Fundamentally, ERS is a site investigation and remediation contractor. And a few points I want to make, though, from this slide. One, we're a service provider, so we don't offer one particular remediation technology or one particular site investigation technology. It's very much about finding the right solution for an individual project. That's very much what we do. And so qPCR, which is obviously what we're talking about today, fits quite nicely with that, because it's a tool in the toolbox that we can use to help make decisions on, is this the right technology to use, and then obviously we can optimize it and monitor that technology goes through. So that's a key point there. The other one at the top there, innovative technologies. qPCR is not that new, but I think certainly some people, it still is an innovative technology, and ERS is always interested in innovative technologies and using them to help support the remediation and site investigation activities. So just a couple of points there. Obviously, site investigation remediation contractor, you can see here sort of the wider range of services that we offer that come underneath that, and I just want to highlight a couple of points here. Obviously, there is that soil and groundwater remediation service offering, and the qPCR helps us in that offering of that to our clients. So we'll be touching on some case studies on, as I said already, on that. And then lab and sort of technical services. And ERS has had its own internal laboratory for quite a few years, and we use that for treatability testing to support our remediation activities. But more recently, it's also looking at providing sort of testing, specialist testing services, and certainly qPCR is a sort of specialist testing service. So offering that directly to clients, and a couple of the case studies fall into that category that I'll come on to, where essentially we've done the testing for external clients for their projects. So moving on to qPCR, and before we get onto qPCR, it's really starting in thinking about DNA and technologies or technique in terms of molecular biology, using molecular biology technique to understand about microorganisms in the environment. And what we can do with molecular biology techniques is we can extract the DNA from a soil sample or a groundwater sample, and then we can interrogate that DNA, because that DNA will represent the microorganisms that are in that sample and other organisms potentially, so bacteria and fungi, other microorganisms, and we can then focus in and ask questions of the DNA that we're particularly interested on, and I'll explain more about that as we go through. So the advantages of this type of approach, of a molecular biology approach, are a couple of key advantages. One, it's quite quick. You're not having to grow microorganisms in the laboratory, which can take time if they're very slow growers, so you can extract that DNA, do your analysis directly on that, and obviously then generate results quite quickly. And associated with that, there are microorganisms, the majority of microorganisms in the environment we can't actually grow on media in the laboratory. So it's estimated that it's probably only about 1 to 0.1% of microorganisms in the environment that we can actually culture in the laboratory. So these molecular biology techniques are really advantageous in that respect, because we're able to analyze the microbes that we can't grow. So qPCR, and these molecular biology techniques are used in a wide range of industries. Obviously in COVID testing, qPCR has been used, but it's medical, it's food, oil and gas, all of these other sort of industries essentially are using qPCR as an analytical technique, and oil and gas is perhaps one of the closest to this in terms of interest in that sector around things like microbial induced corrosion, so looking at sulfate reducing bacteria and being able to analyze them, and in the case of qPCR, looking to quantify them to answer questions. So moving on, first, before coming on to qPCR, just explain about what PCR is. So you can see at the top there, polymerase chain reaction. It's basically, we have this enzyme, this DNA polymerase, that copies DNA. So we've extracted the DNA from a sample, as you say, and now we're interested in copying. And in a very basic analogy, if you think of a photocopier, you maybe have a book, you've got a page that you're interested in, and you want to copy that. In the same way, we can target, if the DNA is like the book, we can say, well, we want to copy that specific bit. So we're interested in the bacteria, for example, or interested in specific degraders. So we take that and we can copy those specific things. if they're present there in the environment. And so PCR is used in a wide range of molecular biology techniques, and qPCR, as I'll come on in a minute, is one molecular biology technique. So the analogy with the photocopier stops at this point, and this image on the right here, I suppose, helps to explain the chain reaction part to PCR. So if we've got the DNA and the bit that we're interested in is in the sample, so maybe it's bacteria, and we want to quantify how many bacteria are there. If there are bacteria there is the DNA, we can then copy it. But what you'll see is as you start to go through various cycles, we've copied that DNA, that then becomes a template. So you can see as you go through the cycles, the numbers of copies that we have massively increases. So that's obviously very different from a photocopier, which is just obviously copying from that single original. Here, once you've made a copy, that then can become a template for further copying. So at the end, you go through a number of cycles, and you can see at cycle 36, you've got huge copy numbers based on what you had originally. So yeah, moving on to the quantitative PCR side of things, then it's very much sometimes also referred to as real time, because essentially that cycle, that copying, we're following it as it happens over time. So if you look at the graph at the top, you can see on the x-axis we've got cycle numbers like we had in the schematic on the previous graph, and then if we have a reaction tube, and in that is the DNA that we're interested in, and then it starts to copy, then you'll get this sigmoidal curve that will start to increase. And the way that we measure that is by measuring fluorescence through some chemistries that I'll come on to in a minute. But obviously how quickly that curve takes off is related to how abundant the DNA you're interested in was in your original sample. So the software then can calculate essentially what we call a cycle threshold, that blue horizontal line on that top graph. It determines for a single reaction, yes, there is that DNA in that sample. That's different from my background effectively. And then it can identify what cycle point that was. So if you then build a calibration curve with known amounts of your DNA of interest, you can obviously, like in a chemical test, you can test your unknowns, and that way you can work back to how many copies were in your original sample. So that's kind of how it's working. It's a molecular technique, but it's a quantitative technique. So at the end of it, we get a number, and we can get a number of how many bacteria in a sample or how many of a specific degrader were there. So a couple more points to make about qPCR, different types of targets. So if you're engaging with a testing laboratory, we talk about taxonomic targets, and a good example of this is a 16S ribosomal RNA gene. So this is fundamental to all bacteria, and this gene is involved in essentially in protein synthesis, so this is why it's fundamentally there. And within that gene, we have regions that are what we say is highly conserved. They're there, the sequences are very common. And so this is a really good target for us to be able to answer those questions like how many bacteria are in my sample. So that's one type of target. The other is what we call like a functional target. So you see the example there. It might be some kind of reductase genes. So thinking about our contaminated land projects, we might be interested in metal contaminant, we might be interested in hydrocarbon contaminants. And at that point, there are only a subset of microorganisms that might be able to act on those contaminants. And at that point, then we can target for specific genes that might be encode for enzymes that would be involved in the breakdown or the change of a contaminant. So that's what we call those functional ones. And then you can build up an array essentially of looking at taxonomic and functional targets depending on the particular project that you're working on, and hopefully that will become clear as we go through case studies. I only touch on this very briefly about the chemistry. So I mentioned in the previous slide that we're measuring fluorescence, and the way that we're doing that is through one or another type of qPCR chemistries. There's kind of 4 that are widely recognized, but 2 that I would say are routinely used. And the first one, cybergreen, essentially what happens here is we've got this fluorescent dye, and when it's incorporated in that copying into the double-stranded DNA, if you look at the top right-hand box there, we see sort of light coming off. That's that fluorescence coming off as that dye is incorporated into the DNA, and that's what we're measuring to indicate that we've made copies of that target DNA. And then the other very common qPCR chemistry is called TACMAN, and it's just slightly different in terms of how it works. You can see this green circle joined with a red circle, and this red circle is a quencher. It's hampering the fluorescence from coming from the green dye again until a piece of DNA is copied, and you can see that the fluorescence, the green, moves away from the quencher, and again the system measures it. So there are a couple of different chemistries, and just wanted to make you roughly aware of them so that if you're then engaging, as I say, with a service provider, you can ask some questions about these. Okay, so yeah, qPCR is a technology, and there are a number of different assays that can be applied with that technology. So as we said already, it might be to look at total bacteria, how many bacteria are in my sample, and rather than do that with a quantitative, a culture-based assay where we try and grow and then count microbes, we're using this molecular-based technique because we don't need to grow them up in the lab, and many of them can't grow up in the lab. So it gives us a number of how many bacteria in the sample, and then we can put that alongside other assays to look at our particular contaminant scenario. So is it hydrocarbon contamination, or are we interested in methanogenesis, or is it chlorinated solvent site? And I've got a range of case studies that will illustrate how we can apply these assays in combination. So obviously, as a molecular technique, it's evolving over time, and the literature is evolving and the assay is developing, and we've contributed a little bit to that space as well. So some of the papers here using different qPCR assays with different types of sample matrices. So just to make you aware of that as well. So why qPCR? We ERS first used it on a commercial project in 2011, and I'm not saying just because we used it then that that's the reason why you should use it, but it was very clear in that project of how effective it could be at helping to unlock a site. So this was a project that we did for an external client, and yeah, really effective for them to be able to take that forward. So it kind of proved for us what I think we thought would be the case, and some of the other reasons why I think qPCR is very effective, very useful, is that it's quick to analyze. So not only just because it's that molecular biology technique, and as I mentioned already, don't need to grow microorganisms, and that analysis of the data is also fairly simple, fairly straightforward. So this makes it comparable to your sort of chemical analysis turnaround times in terms of delivery. The results are fairly straightforward for clients to interpret, and if they're not that comfortable, then obviously they can be that support as well. But ultimately you're getting a number, you're getting a concentration, which is telling you how many microbes or how many of a specific degrader there are in that sample. So I think people, if they can work with quantitative chemical data, there's no reason why they can't work with this type of data either. It's cheaper than DNA sequencing. So DNA sequencing is another molecular biology technique. It's a very powerful technique. So going back to that sort of 16S gene, this gene that's very the fundamental in all microbes, we can actually look at not just the conserved regions, but the variable regions. And by doing that, you can identify the microbes there. But I think it has a much bigger sort of, needs much bigger sample numbers to get that economy of scale. With qPCR, I would say most contaminated sites are that bit smaller. There may be a few borehole locations to maybe a dozen or so. That would be, I think, the majority of contaminated sites, at which point DNA sequencing type approaches can be quite costly if you were to sample and analyze for that sort of number of locations, and it really works more cost effectively with much larger sites. The other point, the difference, I suppose, with this sort of DNA sequencing and qPCR, so with microbes, thinking about bacteria, many of the genes that are involved in specific stages of breakdown of contaminants, those can be found on what we call mobile genetic elements. So these bits of DNA can move between species of bacteria. So with that DNA sequencing approach, if you only look at the 16S gene sequences within a sample, you might not have that understanding of which ones are actually involved in the degradation because of this sort of movement of degradative and catabolic genes. So another reason why I think putting together a few assays and doing qPCR, where you can say, well, this is how many bacteria we've got, and this is how many degraders we've got, can make a more cost-effective kind of application in terms of the molecular biology techniques for contaminated land projects. So moving on to the case studies. So this first one, this was for an external client. The consultant, large international consultant that we were working for, the problem, ammonia contamination in groundwater. This site was near London, so the regulator was the Environment Agency on this project, and really they were asking the consultant, the client, for further evidence. So they were putting forward this idea that the ammonia contamination was being acted upon, and you can see there the proposal that there's sort of ammonia oxidation as a primary pathway for monitored natural attenuation. This was the proposal being put forward, but the regulator saying, give more evidence that might be the case. So they came to us to help with that in terms of providing that qPCR testing service, and we looked at that in terms of testing samples for them. So the graph here, you see 6 different samples just labeled here 1 to 6 for simplicity, and then on the right hand side on the y-axis you've got gene copies, and we applied 3 different assays to the samples. So the blue vertical columns, that is the total bacteria within the samples, and then we've got the orange columns, which is ammonia oxidizers, and then we've got NARK, the gray columns, which is nitrite reducers. So looking at a couple of different parts of the nitrogen cycle with those maybe what we'd call the functional assays, and the total bacteria being that taxonomic assay. And if you look at the results, so we've got consistent sort of concentrations of total bacteria in samples 1, 4, 5, and 6. But when you look at the ammonia oxidizers, you can see in samples 1 and 6 that these are elevated relative to 4 and 5. So with the horizontal grey lines, if we go from one grey line to the next, this is an order of magnitude increase. So we're seeing some big changes in ammonia oxidizers in 1 and 6. So as a part of the total bacterial community, essentially we're seeing more ammonia oxidizers in those locations. And this was really useful data for the client and consultant, because those locations corresponded with the chemical data to help them build that case, essentially. So adding what we call that additional line of evidence to support that proposed biodegradation pathway, that we've got ammonium oxidizers that are being enriched by that contaminant being present. They're acting upon that, and that's supporting that M&A approach. And through this qPCR data, with obviously the work that consultant did, that helped to satisfy the regulator and take that development forward. So second case study, so this was a chlorinated solvent project and an active remediation project and a project for ERS. So ERS was involved in delivering the remediation on this project. A former manufacturing site in England, as it says there, chlorinated solvent contamination in the groundwater. We didn't have qPCR involved in this project at the very beginning, but the strategy was to apply bioaugmentation, so adding a culture along with reagents that would support that culture and other microorganisms in the environment to really perform an reductive dechlorination. So if you look at the schematic at the bottom there, I'm sure most of you will be familiar with this approach of detoxifying chlorinated solvents like PCE and TCE through substitution reactions of the chlorine with hydrogen. And there are certain microorganisms that are really good at that. So things like D. halococoides is able to carry out that process all the way through to ethane, where some other microorganisms might only be able to do some of the initial stages. So the culture that was used had D. halococoides in it. It was a mixed culture, but certainly D. halococoides was a key component of that bioaugmentation culture. And so ERS introduced that culture with those reagents by direct injection. And this is just some of the data from that site, again, the qPCR data. So we had a consultant taking samples and sending to us for qPCR testing. And this just shows the D. halococoides specifically within that bioaugmentation culture. So the pre is before we've put that culture in. So there was actually D. halococoides already present in the environment, which is not that uncommon. But obviously then following the introduction of this bioaugmentation culture, we see, as you would expect, increases in the abundance. And then we're able to track and monitor over time how that culture was maintained, potentially stimulated as it acts upon the reagents that were put in with it. It is a slow growing microorganism, but you can see the data here. And as I say, this qPCR wasn't part of the original plan for this project, but the consultant, the client, became very keen, interested in this data as the project evolved, and certainly helped them to put the case to the regulator again in terms of closing out this project. So third case study, again, a chlorinated solvent one, but this time looking at using qPCR a little bit more front end, so helping with selection of a sort of remedial design. The site this time, a former tannery, so the historical use of the site using chlorinated solvent as a degreaser, and you can see on the map there the former footprint of the degreaser building. It was being redeveloped for housing, so needing active remediation to fit in with the time plan for that site. As is the case, I'm sure people will be aware with chlorinated solvent or DNAPL site investigations, it can be quite complicated. And certainly in our proposal for this project, there was a plan to do some additional site investigation because it's quite difficult to understand perhaps what the extent is. So before applying that active remediation, working out exactly what the injection plan was going to be, it needed a bit more site investigation work. And so as part of that, we did some qPCR analysis, and particularly looking again at sort of degrader abundance. And as it says there was very low degrader abundance in this site, so much less than we saw, for example, in the previous site. And this data then helped us to formulate what approach to apply on this site, what reagents to use, and we decided on a combined biological and chemical reduction approach on this particular project. And made real use of the boreholes for that additional site investigation by injecting the reagents via those boreholes into that site. And in terms of the project, so it was effective in terms of the reagents of creating this negative redox environment to stimulate the reduction of the chlorinated solvent. And then we also saw the maintenance of the degraders over time as well, and saw significant mass removal in this project. So even though this project was one that fell during COVID, the impacts of COVID, and as I mentioned, as housing development, we're still able to meet the original time frame of the project. So successful project in that sense. And the fourth, just conscious of time, the fourth and last, I think, case study, this time looking at petroleum hydrocarbon. And this is for, again, another for a consultant client rather than one of our own projects. So a fuel filling station where they'd had petrol leak, and there was ongoing active treatment. So you can see there sort of air sparge and SVE treatment, and obviously the costs involved in running that and the benefits that gets over time. The consultant and the client were looking to transition from that active treatment to a passive treatment. So again, looking for evidence to help support why that might be the right time or an appropriate thing to do to move on to monitored natural attenuation. So our original project, so again, we weren't at the front end of that project. There was obviously that active remediation going on, but the consultant looking for a bit of additional data to support this strategy. We initially proposed to do a couple of assays, the total bacteria, along with this ALK-B, which is focused on alkane degradation, so a gene involved in stages of alkane degradation. And what we found with the data was, well, we got the results for total bacteria, but there wasn't much in terms of abundance of ALK-B genes in this environment. And there may be a lot more going on behind that, which in terms of the particular assay that was applied to that site. But what we were able to do was quickly add in some additional assays, which again sort of I think shows the flexibility of qPCR as an approach. Once you've extracted the DNA, then you have got this sort of suite of assays that you can apply. And we added in some assays, having reviewed the chemical data with the consultant, and these assays were looking at enzymes, genes, encoding enzymes that break down aromatic compounds, which was another key contaminant of concern in this scenario. And that was very useful for the consultant on this project in terms of helping to build up a better, stronger picture of what was going on in this site. So yeah, I think illustrating the flexibility and the quickness of being able to turn this type of data around. So the client very happy with both of those things and helping to support that strategy to go forward. So I'm just conscious of time. I will skip over this last one. Just quickly on the workflow. So early engagement, I think, is really helpful in terms of selecting which assays to apply, whether you're applying that on all locations or whether it's much more focused in terms of that spatial arrangement, helping with that. Obviously helping with sampling and curing of samples. Then UK-based service, so obviously being able to take those samples and process them quickly and turn those results back round. And then obviously in terms of results interpretation, helping clients as and where needed. So just quickly to summarize, I think it's a technique that not the whole of the UK contaminated land industry is aware of. So this has been something of a sort of passion of mine to try and raise awareness of the technique with people. And one of the reasons why I'm thankful for the opportunity today to tell you a little bit about it. It's got a demonstrated track record as a line of evidence, and I think I've given examples of that with different UK regulators where it can help to support projects move forward. Fundamentally, if you're not sure where you want to apply it, it's really thinking about scenarios where you're interested in, as it says there, microbial driven processes. So, you know, biological, microbial degradation of a contaminant. Also impacts on microorganisms in the environment. So a contaminant having an impact on microorganisms. Equally, you could use technique in that kind of scenario if that's of interest. It's not beneficial to all projects, and as I mentioned, ERS, as a service provider, this is a tool in the toolbox for us. So we're not going to force it on you. But it is there to uptake, and really just want to make sure that it is used effectively. So making sure that that design does give maximum benefit, but control budget at the same time. So we're not coming to you and say, well, you need to test all samples all over the site. No, let's come up with a really sensible design that will give you the data that you need in a cost-effective way. So I think that's me.
That's brilliant. Thank you very much, Tom. I think we could have given you a full hour slot, I think, because I think there was lots more that we could have tried to get in there. But I think it's given everybody a bit of a taste for what the techniques can be used for. Everybody's a bit quiet today. I don't know if they're sort of like already in holiday mode, but don't have any questions at the moment. I'll just ask you one, and then we can sort of move on to Gareth. I wonder if you could maybe just sort of outline what the sampling requirements are in terms of amount of sample, any specific requirements in terms of transport or preservation or anything like that.
Yeah, I mean, it's not too dissimilar from how you would go about with a chemical testing. We can provide bottles for people. We use ones that are virgin and sterile, obviously given the nature of the type of testing that it is. And we essentially can send those out to clients, and then they can send them straight back. Then it's about keeping the samples cold, as you would standard practice with your chemical testing. And also, if most of you've seen the case studies here, it's applying it to mostly groundwater samples. So filling that sample up to the brim, really to help make sure that there aren't the changes with that. But obviously, as I mentioned, we have that UK base, so it's good for us in terms of being able to get samples quickly and then process them. And once then we've extracted the nucleic acid, then taking on from there.
Brilliant. So as we're a wee bit over time, I think we'll probably just leave it there at that. If anybody does have any more questions, I'm sure Tom will be happy to pick them up later on. I know he is hanging around, or we can sort of do them offline. So thank you very much, Tom. That was great, and I'm glad we managed to finally hear your presentation today without any glitches, hopefully, for everyone. So I'm just going to invite Gareth now to the stage. I'm just making a presenter. And delighted to have Gareth Leonard here, who's a remediation manager from Regenesis, managing director for Regenesis, and he's going to talk about the use of colloidal activated carbon for in situ remediation of PFAS impacted sites. So I'll just hand over to you, Gareth.
Oh, there you go. Can you hear me now?
Yep, we can hear you. We just need you to be full screen on your presentation and you'll be good to go.
Full screen on presentation.
Just in your presentation view.
There we go.
That's you, super.
Thank you for bearing with me on that one. Okay, let me just clear this other screen that I'm looking at as well. Okay, so I'm looking at doing about 30 minutes on this. Again, it's going to be a bit of a ride through. There's a lot to talk about here, so I'm not going to be able to touch on everything, but hopefully we can answer some questions afterwards or provide some information after that. So I'm going to talk about the use of colloidal activated carbon or liquid activated carbon for the in situ remediation of PFAS on PFAS impacted sites. Okay, so I'll talk about what the technology is and the practical approach on site, including something we call DVT, which is checking the location that the treatment is going to take place. I'm going to then talk about plume treatment and give you a couple of case studies on that, and then talk about source treatment. In my mind, that's slightly the wrong way around, but the reason I'm doing that is because we had a plume technology that we discovered we could use for PFAS, and since then we've developed a source technology which isn't as developed, so I'll show you where we are with that, and I'll show you what further work we're doing. So that's why it's a little bit the wrong way around, this presentation, but hopefully I can just tell you where we're up to. So this is the typical sort of result you get with a colloidal activated carbon treatment of a PFAS plume. You inject the product, you sorb the contamination, and you go down to very, very low concentrations. Notice the y-axis is in nanograms per litre. So this is our site in Ontario. It's been going on for 6 years with those concentrations there. But I'll show you a couple of sites in the UK. So what is colloidal activated carbon? We've taken granular activated carbon, which has a diameter of anything from 1,000 to 1,500 micron in scale, and we've ground it down to the size of a red blood cell, to the size of a single microbe. So that's about 1 to 2 micron. We've then suspended it in water with dispersion agents, so they don't clump together again, to create a true liquid. It's like an ink, it's like a dye. And the idea is you inject that into the subsurface under low pressure, so you're not having to fracture it in, because the particle size is so small, it just flows through pore throats, and as it moves through those pore throats, it crashes into the aquifer material and stains it. It coats the subsurface in activated carbon, creating a subsurface filter. So you see some scanning electron microscope pictures at the bottom there. The scale's probably a bit too small for you to see, but some of these gaps are 10 to 20 micron. And then we've zoomed in a little bit here, and you can see after we've applied the colloidal activated carbon, you can see individual particles of about 1 to 2 micron. So this gives you extremely fast sorption of PFAS. The smaller the particles are, the more there is the exterior surface faces the PFAS that allows sorption to occur. And also there's a shorter distance to those sorption sites within the activated carbon itself. So you get very efficient treatment, you get very effective treatment, and there are studies to show that the smaller the particle is, the more effective treatment you get. The other thing is, because of the small particle size, we're not affecting permeability. This is what the product looks like. It comes as a concentrate. You dilute it a bit further on the site and then inject it into the subsurface. You can see how easy and straightforward that is to inject. The trick is just getting in the right place. So on the left here, you see colloidal activated carbon, and on the right, powdered activated carbon. And what was happening there is we're just flowing it through some silty sand under gravity. And you can see that the colloidal activated carbon moved nicely through that silty sand, and it's left a gray color. So it's coated the aquifer material in activated carbon. And we're able to go into these tests and work out what sort of coating is left. And essentially it is a single coating. You're getting a 1 to 2 micron coating throughout the aquifer, so you're not affecting the permeability. You want the groundwater to continue to flow through these barriers, these permeable reactive barriers, these treatment zones, but the PFAS will be sorbed. You can see on the right, the powdered activated carbon. What's happening is it's clumping, so it's maybe 20, 50 micron in diameter. It will be clumping to create larger particles, essentially of about 1,000 micron, and they're just not getting through the pore throats. So to get that in the ground, you would need to do high pressure fracturing, and then you're not getting it into the flux zones. You're not getting it into where the contamination is flowing through the aquifer. That's the zone that we need to target. So this is a typical site that we might be looking at. Here it says IPRB, so that's injectable permeable reactive barrier. What we've got down here is a fire training area. It's where they've sprayed AFFF, so aqueous film forming foam. There's a big move now to move to fluorine-free foam, or F3, but there is either foam still being used, or there is the residual contamination from the use of this foam over many, many years. The PFAS from the AFFF gets into the groundwater, and then it's highly mobile. It's very soluble, and it moves through the groundwater and flows off-site. So you're getting an off-site liability for the site owner. and then you are potentially impacting the aquifer itself, drinking water, surface waters, etc. So the idea is that we come in to the edge of the site typically, and we would inject a series of injection points with overlapping radius of influence, so not quite as the animator has made here, so there's no gaps in between it. And what happens is the groundwater continues to flow off the site, but we sequester the PFAS as it moves through that zone. So what we're doing is eliminating the risk from PFAS to the down gradient receptors. So as you know, risk is hazard times exposure. We sequester the PFAS in situ, so we eliminate the potential for down gradient exposure, which eliminates the risk. So this approach is being accepted, increasingly being accepted. We started working on this 6, 7, 8 years ago, potentially, and regulation around the world is in different stages. You know, there's a lot of research going into what we should be doing with PFAS. But coming from the States, there is increasing regulation talking about what they call enhanced attenuation. So essentially, you're looking at what we're doing here. You're either reducing the source, the discharge from the source, you're preventing the movement of the contamination away from that source, and you're allowing attenuation of the residual plume, much like we do for many other contaminants. And the difference being that PFAS doesn't degrade, but this approach is still done for the likes of metals, etc., where you might be doing stabilization, etc. So it's a similar sort of approach that is beginning to be accepted around the world by different regulators. And it's very patchy. We're doing work in certain countries, and it's not happening in other countries. Some countries seem to be ignoring it. Some countries seem to be working towards targets. Other countries are taking a more practical approach. So the ITRC is saying groundwater containment may be achieved through the injection of amendment that causes PFAS to attach onto materials in aquifers. So that's what we're doing. This is an interesting paper that's come out. It came out as 2 papers about monitored natural attenuation or enhanced attenuation. And it's by Chuck Newell and his group that are essentially working for the EPA, trying to work out what can be done with PFAS, and that can then guide the remediation industry, certainly in the US, as to how that we can deal with it. And of course, one of the concerns is who's going to pay for all of this, because PFAS is you get gigantic plumes. They're way beyond the polluter's site itself. So how do we deal with this without nation states taking on all of the cost for that? And I think you can see that with the regulation coming through in the States from the EPA, that you're going to be dealing with the source area to the edge of the site. In the US, probably in the UK, Scandinavian countries will probably push out treatment way beyond site boundaries. So anyway, adding sorbents to increase retention is seen as a remedial solution. Does it treat everything? Well, I'd love to say it does, but there is, of course, a treatment envelope to every technology. Looking at the plume and looking at the type of contaminant, we can treat C6, C8, the contaminants that tend to attract the remedial targets on sites. C10, C12 and above, they tend to be already sorbed to the soils and they're not moving anyway. So it's these problematic compounds that we can target. C4 and below are challenging. You know, we will see reductions in them, but they're not particularly sorbent. They're highly mobile. They're not attracting remedial targets at the moment. That is something that toxicologists are working on. Concentrations, so we have a wide range of contaminant concentrations that you can target with activated carbon. At low concentration, we can get to non-detect. That is the real benefit. The great thing about using the small particle size and using it in situ is that you get really efficient sorption, and you're getting a slow groundwater flow, so you're getting a lot of retention time in the subsurface compared to using granular activated carbon in a fast flowing pump and treat system ex situ. If you're getting really high concentrations, you're getting into the source area. I'll come on to source area treatment. You might be looking at a different approach. Groundwater flow, of course, is important as well. We're wanting to look at the flux of the contamination coming into the barrier. So that is that considers groundwater flow velocity and also the level of contamination in that groundwater. So if you've got low groundwater flow, absolutely fine. If you've got really high groundwater flow, you might be struggling to retain the contamination. You might get breakthrough, and it might be something that we might struggle with. So in terms of longevity of treatment, a typical contaminant would degrade, a typical organic contaminant would degrade. So we'd get absorption component when we're designing these sites, and we'd get a degradation component. With PFAS, currently it's understood to be difficult to degrade, although there are studies where they're looking into potentially there is some degradation going on. There's some promising work from Stanford on acidimicrobium difficile that may be having something, but we're perhaps a way off from that. So the modeling that we do, the design that we do, is based on dynamic sorption. So that's sequestration of the contamination as it moves into the barrier, and further contamination comes into that barrier. So what will happen is the barrier will eventually become saturated over time. So how long are we sorbing this? How long are we retarding the contamination for? And it's extreme retardation. A natural retardation factor in a natural aquifer might be something like 3 to 10, and that's the groundwater velocity divided by the contaminant velocity through the subsurface. With an activated carbon, you're looking at something like 1,000. So you can actually sequester for decades, maybe even longer. And what you've got to look at is your target contaminants. You've got to look at competitive sorption from chlorinated solvents or TPH in the site, and natural organics that are coming in as well. Of course, these will degrade. So what happens if you do start to have breakthrough, or if you're planning to have breakthrough, because we can model when that will occur? Well, of course, you can reapply. And I'll come on to this, but the benefit of this approach is that you inject, it just takes several weeks on site. It stops the contamination going off the site almost immediately, and that's it. You just have to monitor every year, or whatever is agreed with the regulator, for decades, until you know the breakthrough is going ahead, until the monitoring showing that's going to occur, and then you come back to site and you reapply. So there's no contamination coming to the surface. There is no energy being used. It's a very passive, sustainable, low impact approach. The longevity, of course, can also be extended by source treatment, and I will come on to that. We can provide options there as well, but of course there's very simple things that can be done there as well. So barrier maintenance, here's an injection. We've got the contamination moving through the groundwater. We inject, we're preventing that contamination going off site. In 50, 60, 100 years time, we start to see low concentrations of the shorter chain material starting to increase, and that's the canary in the coal mine telling us that the problematic compounds may start to come through soon. So we come back to the site and we reapply at that point. And you're not taking up huge amounts of the site. These injections just take up a couple of meters on the edge of the site. And then you get another 60, 100 years of protection of the environment from that. So very low impact solution. But of course, as I mentioned, and I'll come on to, what you could also do is reduce the discharge into the groundwater, and then your barrier that would have lasted 100 years then may last much, much longer, and you may not need to reapply in the future because you've reduced that influx into the subsurface. There's a really interesting article that's just come out I wanted to share with you. This is from Grant Carey and his group, often working with the University of Waterloo. And they looked into the longevity of these approaches that we've completed. So they looked at 17, they did many things, I'll come on to them, but the first part of the paper is they looked at 17 of our sites where we've got data up to 6 years so far. 14 of these sites, we were showing about 90 to 99% reduction down to essentially non-detect. One site was showing variable results, 82 to 99%. And one really important thing we found in all of this is you're dealing with really low concentrations, and it's really important to know which way the groundwater is going at the point that you're treating it, particularly when you're doing a pilot that's sort of 10 meters wide. And what we had on this site, as I've seen on others, is if there is seasonal movement of the direction of the groundwater, then you can get bypassing of that barrier. So it's crucial that a really good understanding of what the groundwater is doing, what the flux zones are vertically, what the direction is that it's going in, is gained before you put in the barrier. And so that's why we're seeing sometimes that we're getting a bit of a bypass of this barrier. The final site was stopped. Basically, it had a huge amount of natural organics on the site that was blinding the treatment, so it's not appropriate for that site. What they then did is they then decided to come up with their own models of longevity. So they created forensic isotherms for PFOS, P4, perfluorohexane sulfonate, and 6,2-fluorotelomere sulfonic acid. They took water from an AFFF site and our colloidal activated carbon, and they worked out the sorption isotherms for those contaminants. They then modeled longevity, taking a typical design of a colloidal activated carbon barrier, and then taking these isotherms and putting in it. And they chose P4 because it's the least sorptive of those contaminants, so it would give you the most conservative result, the quickest breakthrough. And they modeled that they would get breakthrough between 66 and 265 years from a single application. So for the other contaminants, it would last even longer. They did sensitivity analysis, and what they found was really driving sensitivity was the dose that we put in, and also the influx. So that's a function of the groundwater velocity and the PFAS concentrations that are coming in. Interestingly, they didn't find that the longevity was particularly sensitive to the target. So if you've got 65 nanograms per liter versus 40 nanograms per liter as a target, essentially, in 260 years, it might be the breakthrough is in 264 years compared to 267 years. It's really you've got to get the design right for the influx to give you that longevity. Another thing they did is they studied 96 PFAS impacted sites. 82% of them showed PFOS and perfluorohexane sulfonate on the site being more than 70% of the problematic PFAS compounds on those sites. So most of the sites were mostly comprised of PFOS and PFHXS. And the great thing about that is it sorbs readily. We can get really good longevity from that. They also said in their review of the technology that preventing and reducing flux coming from the source increases longevity. So I'll come on to that. Okay, so let me give you a couple of examples from the UK. I am running late, so I'll be quite quick on this one. So this is the first site that we did. It was a private UK airport in England. Contamination coming from a fire training area. They're divesting the site. Fairly low concentrations of PFOS and P4 was what was driving the risk. That was what we had targets on. It was in a weather chalk, very slow groundwater flow. We identified an area to do the pilot study on. We then put in a line of treatment. Where's my pointer gone? So a line of injection points with groundwater moving from down gradient through the barrier, moving from up gradient through the barrier to down gradient. And here's the results. So we've got the up gradient wells, shallow and deep. Then we've got the in barrier well, shallow and deep. Ignore this one for now. Then we've got the down gradient well, 9, shallow and deep. So what you're seeing is after injection, we get immediate reduction to non-detect. And this is for PFOS and P4. I will show you on the next site full PFAS results as well. And then down gradient, we're seeing a reduction, but not an immediate reduction. And it's only 5 meters down gradient. And this is another interesting learning point. If this was a petroleum hydrocarbon site, you probably see the results come down quicker because you're generally dealing with higher concentrations. What we've got here is nanograms per liter of contamination. There is contamination in the immobile porosity down gradient. So what happens is we create the barrier, clean water is now coming through, but you have to wait for back diffusion of the down gradient contamination to become spent. So you see this happening slowly over time, so it can take a bit of time down gradient. So what we did is we went back to the site and we put in another monitoring point in between the barrier and the down gradient well, just to see what was going on, and we were at non-detect there. So that clean wavefront is moving through off-site. So we got a 99% or more reduction in PFOS and PFOA, clean water's coming through. We moved to full scale and we injected that finishing this summer. It's a 277 meter long barrier. Concentrations are now below the target, which is 0.1 microgram per liter for PFOS and PFOA. And we've provided a warranty on that site. Should there be any issues, we'll just go back and sort that out. The site that we're working on right now, this is an international airport in the UK. Again, fire training ground. We've had to blank it out because they get very paranoid that people can recognize each other's fire training grounds. The fire training guys or the firemen, fire people can recognize the different shaped pretend planes that they have. So we have to cover them over when we're talking about these sites. There's a known PFOS issue. They decided to voluntarily clean up the site because there's a triple SI just off site. What we've got is alluvium with slow flowing groundwater, river terrace gravels with quicker flowing groundwater sitting on top of London clay. Groundwater is about 2 meters below ground level. Total PFAS is greater than 200 microgram per liter. And we've got petroleum hydrocarbons on the site as well. And you see that a lot as well in fire training grounds because they're splashing fuel around to get the fires going, etc. So you do see a bit of TPH. So one thing we did first, and we do it on all the sites, is design verification testing. And we'll either do it or the consultant will do it with us advising or a bit of both. And what we really want to do is understand what is going on within the treatment zone. So we're really looking at where that flux is moving through vertically, what direction the groundwater is going, what speed the groundwater is going at. We want to know the silt content of the different layers so that we can know how the colloidal activated carbon will adhere. We'll do permeability testing. We'll tend to use iFlux when we've identified the flux zones to see what's actually happening in those flux zones. We might look for extra contaminants. We might look for the hidden PFAS using top assay. We'll then do clean water injection testing to see what we can achieve. And then we'll do a pilot using colloidal activated carbon. And what we're looking at doing is getting interlocking radius of influence. So we'll then sample the groundwater away from each injection location. And here you can see we're doing some colorimetric testing to see that we've got the right concentration of colloidal activated carbon at the right distance so that we've not left with holes in the barrier essentially. Okay, so the first thing we did on this site is inject a parking agent into the river terrace gravels lower down, further down gradient, in the pilot area. And what this is essentially is a flocculant that breaks the dispersion agents of our colloidal activated carbon. We don't need it in the upper slower zone. You put in the right dose, it moves through the groundwater, it coats the aquifer and it's spent. So we get the barrier design we want. In those river terrace gravels, it's going quite quickly. We don't want to lose the product. We don't want to have a sort of a weak barrier over a greater distance. So what we can do is we can engineer the shape of the barrier using this parking agent. So we injected that, then you inject the colloidal activated carbon into the subsurface. So then if we look at these results, these are the up gradient wells. If you see here, up gradient, and there's the up gradient results. We're now in barrier there, down gradient, and then down gradient deep and shallow. So if we look at the results, if we start here with the in barrier, these are the baseline we inject. we get a reduction in the contaminants. Here we inject, we get a reduction in the contaminants, and then that's seen down gradient as well. Up gradient, you see it deep, and this is because it's much more permeable and it's quite close. So our radius of influence is actually going about 2.5 meters, which was a bit more than we thought it would, but it's okay. You don't see such an effect in the less permeable upper zone, which is to be expected. So we're getting a 93% reduction in some 11 PFAS, and in the target contaminants P4 and PFOS, we're getting down to detection limits. So we've now moved on to full-scale treatment. We're injecting now. It should be finished. It should be finished tomorrow, and it is 76-meter extension. So basically, we've got the pilot section already put in. Okay, it's a weird shape, but we've got that preventing contamination going off-site. We're now stitching in the rest of the barrier along the edge of the site. We're putting in a Petrofix barrier here as well. And Petrofix is just another form of colloidal activated carbon that have nutrients in it for petroleum hydrocarbon remediation. And that is next to the interceptor that is leaking a petroleum hydrocarbon. So we want to tidy that up as well. We've then put in some extra upgradient. The consultant has put in some extra upgradient monitoring locations in barrier wells and down gradient locations. And all results are looking good so far. So I am running out of time, but I'll go for another 3 minutes or so. Source treatment, I said, was important as well. PFAS, when you spill it, hangs up quite high in the vadose zone. It sticks quite nicely, but it does then infiltrate through due to rainfall and get into the groundwater, and you can get this reservoir of contamination that drives risk for a very long time. And that's because you don't need much of it to get in the groundwater to exceed the targets that are starting to appear. So this mass can be a problem for a very long time. What you can do is you can mix activated carbon into the upper portion of the vadose zone, and that reduces the leachability of the contamination that is on the soils. You can change the permeability of the formation as well with the cement. Cement's not so good at actually binding PFAS, but what you're doing is you're preventing infiltration, so that prevents contamination moving through. Those two things, the leachability works better on the longer chain material, the reducing the permeability prevents the shorter chain material being washed through as well. And there's a site that we've done this on in the US. We've only done this on one site so far. To be fair, we've got another site planned in January or February. We have another colloidal activated carbon product, rather unoriginally called Source Stop. As we had Plume Stop, we got Source Stop. And what you can do with this is you can put it into the base of the excavation, or however you're doing the in situ treatment, underneath your vadose zone treatment. And what that does is it moves through the vertical pathways, coats the residual contamination that's in the subsurface, and prevents any further contamination that is infiltrating through. It will catch that and prevent discharge to the groundwater, so that the down gradient plume can then just attenuate. And there it is being put in the ground using drain pipe technology. You can then inject it into the subsurface as well. PFAS is surface activated, so it wants to hang around at the air-water interface, so you'll get a lot of it in the capillary fringe. You'll also then get a lot of it just underneath the source area in the groundwater. So you can inject this colloidal activated carbon just in the same way as I was talking about before, but instead of a barrier, we're targeting all of the contamination in this zone, and you just sorb that contamination and prevent that from then moving further down gradient, and you're essentially cutting the head off the plume and allowing that to attenuate. So you can combine these two things, and I've put this in a certain order. What you tend to do with in situ is you are causing disturbance when you're working, so the thing to do first is make sure the gate is shut. Okay, so you work yourself back into the site, typically on a site. So here we're closing the gate, we're putting a barrier in, there's nothing further going off the site. We then move into the source area, we inject in the source area, we target that capillary fringe, we get rid of we sorb the contamination that's in the source area, so it's not moving down gradient, and then you move into the vadose zone and stabilize that, prevent that from leaching into the subsurface. Or you can cap it. A lot of these sites, if they're not an airport, if it's in the middle of town, etc., make sure the drains aren't leaking, replace those, cap the site, make a car park. If there's no groundwater coming through, the PFAS that's located in the top couple of meters is no longer leaching down into the groundwater. So there are engineering solutions as well that are compatible with what I'm talking about. Okay, last few seconds, sustainability. The approach doesn't need pumping, it's not bringing anything to the surface. So we're working with Ramboll to produce some life cycle analyses on not only on the product but on the technique, so that it's becoming increasingly important, the sustainability of these approaches to our clients, particularly large international clients. Here's some results from a study that was done for chlorinated solvents, and you can see the difference between a pump and treat system over 30 years and plume stop in terms of greenhouse gas emission, hazardous waste, etc. So I'm hoping it's going to be a similar sort of results, but we're in the middle of doing that now, and I'm hoping to get some results from Ramboll in February time, something like that. So we've done about 40 sites worldwide. We're looking at a site a week at the moment. There's 8 academic papers not written by us. If you want to look those up, you can go on the website or I can provide them. Ongoing studies, there's a bedrock study at Toronto University. University of Waterloo is doing some isotherm studies as well, such as similar as I showed you today. BioPFAS is an interesting group in Sweden, where they're looking at whether there is microbial degradation going on a well, just in general, but also they're going to be looking at an area where we've injected colloidal activated carbon, see if there's any microbial degradation occurring. We are using colloidal activated carbon to clean systems such as fire trucks, AFFF systems. So we've done a fire truck in Belgium, so we're looking at that side of things. We're doing some testing soon on passive diffusion bags, which you might have seen. They're starting to produce them for PFAS sites, so we're giving those a go. And I mentioned the sustainability side of things. And that's me. Thank you for your time.
That was absolutely brilliant, Gareth. I think we'll need to have a wee follow-up session in the new year, I think.
Yeah. I might need to lie down first.
Yeah, so that was great. We'll take one question just now, if that's okay. So we've got a question from Alex who's asked, if you have the PFAS, etc., sequestered at the injection sites, would the soils at the site become a hotspot of contamination, and what are the options for this? So
that's a question that gets asked a lot. I tried to sort of address this in there. You put in, ideally what you'll do is you'll do everything together. You'll do a source treatment, so you're reducing what's then coming down into the groundwater. And it might be, I can see this in the future, particularly in the UK, that is all that is done. If, however, there is a receptor off-site and you need to protect it, you'll then put in a barrier. And that might be it. The level of contamination in the groundwater is really low. It then sorbs onto the colloidal activated carbon, and it'll, if you have reduced what is being discharged in the groundwater, you may never need to apply any more product again. That's essentially it done, and you're never going to come over targets again. If it's a case where there is an ongoing source that you can't get in and treat that source, and you do put in a barrier, you will in the future potentially need to reapply at that location. And that's because you get rollover, you get the contamination moving through your barrier. Hotspots is an exaggeration in terms of the level of contamination that's in here. It's just that at some point it is going to break through and start to move into the groundwater again. So you can then just reapply a down gradient. You're only using up 1 or 2 meters of the site, so you're just injecting in pretty much the same location, and you might be doing it in 50 years, 100 years, 200 years, something like that. So it is a case that there is maintenance work to be done, but you're talking about a few weeks work every few decades to prevent that contamination moving off the site. But as I say, in the future, it's really going to be that combination of source and plume that avoids any future work being needed.
That's brilliant. Thank you very much. I think we'll just leave it there so that we can quickly squeeze in our AGM before 2:30. So thank you very much to Gareth and also to Tom for today. If you want to watch back, because there's a lot of information in both those presentations, then we will upload it onto our YouTube channel probably tomorrow. Go back and have another look, and we'll probably have a wee word with Gareth and see if there's anything we can maybe do in the new year that's maybe a bit more detailed to give and if you have any thoughts on that, anybody who's in the audience, of what you would like to see in that, then just drop us a wee email at admin@sclaf.co.uk. So thank you very much. So without further